Okay, so let's get started. So, how you doing, sir?
Hi, tired. Many
Tired.
meetings in the morning, but it's okay. It's a difficult time, with all these science going on about the corona and trying to find a solution. We scientists have to work hard.
Yeah, no doubt, no doubt. I think most people right now are in question as to what's going on, but I think lots of people are starting to go out a bit more now. You know, at first it was full on lockdown, and now people are. You'll see them in parks and stuff like that.
I think spring is giving us a break, so we'll be fine until autumn. But what will happen in autumn, nobody knows. So for
Yeah.
now, at least I'm in Croatia. Croatia is fully opened. We are inviting tourists to come. Beaches are open. So
Oh, really?
here it's okay. Yeah. We have
Oh,
0 cases for a week. So we are kind of corona-free country.
okay, okay. Don't say that too much, otherwise you might get a sudden influx of people going to your country.
We'll cope with that.
Oh, that's cool. So, GlycanAge is the discussion today. Now, from what I understand about GlycanAge, they use a patented technology. Um, they also advise that it's the most accurate indicator of biological age test and overall health. Now, I understand that you are a professor of biochemistry and molecular biology. Could you give us a further indication as to what your scientific background is and what you can tell us about GlycanAge?
Okay, so I will start by telling you something about glycans, which are the basis for GlycanAge. So glycans are modifications of proteins which make protein different. So proteins are the molecules which do most of the work in our body. And if you go back in evolution, first we had nucleic acids, then nucleic acids kind of invented proteins to make them more diverse, to do more jobs. And once we became multicellular, so no longer a bacteria, but multicellular organisms, we started to add glycans to our proteins. And glycans are kind of, they're doing the fine tuning of proteins. They regulate the protein activity. And one very important protein is immunoglobulin, antibodies, which actually they are used to destroy all the pathogens which attack us. And the activity of immunoglobulins is regulated with glycans. And I will not go too deeply into that. Maybe later we can go if you want. But we know that these glycans are regulating immunoglobulins, and in this way, they are kind of regulating inflammation. But the problem with glycans is that they're very difficult to analyze. And majority of scientists do not do that. So they just look at DNA, they look at genes, they look at proteins, they don't look at glycans. So I work in the field of glycobiology for 30 years, and slightly over 10 years ago, we started to do large studies of the glycome. We were the first who started to do large studies of a glycome. And the first thing we learned is that glycans change a lot with age. So when people are young, glycans and their immunoglobulins are functioning in a way to suppress inflammation. As we are getting older, from reasons we don't fully understand, glycans are becoming more pro-inflammatory. So old people are generally having glycans which promote inflammation, while young people suppress inflammation. And these changes also happen in a disease. So when people get ill, their glycans become more pro-inflammatory. And this happens in many different diseases. So it's not a disease specific. It's a kind of one of the molecular mechanisms which is contributing to disease. And actually there is even a whole hypothesis saying that the aging is being driven by inflammation. And this is so-called inflammaging hypothesis.
Yeah.
And glycans there play an important role. So once we learned that the glycans change with age, then we tried to predict somebody's age based on glycans. So we just measured the glycans, and it was a couple of thousand people, and modeled it to predict age. And we could predict age with an error of 9 years. which is not bad. You know, you take a drop of blood and say this comes from a 40-year-old or a 20-year-old person. But the most important thing was when we looked at the difference between the real age and the glycan age. So the real age meaning the chronological age, the number of years since you were born. And then we realized that the difference between these two ages can be explained with a lifestyle. So if somebody is living a healthier life, then the glycan age would move slower than the chronological age. So somebody would be 50 and the glycan age would be 30 or something. And if people live different types of unhealthier life, then the glycan age would move faster and you would be 40 chronologically and your glycan age would be in the 50s or 60s. And this often correlates with the disease. So if people have some diseases, the glycan age move faster, but it can also correlate with, for example, obesity is a driver. If you are obese, your glycans move faster, but also it's not only obesity, it can be a different form of, for example, professional athletes usually look older. But you know, if you talk about a football player and he's 34, everybody would say, oh, he's so old, 34-year-old football player.
Yeah.
But from kind of, I know in science, 34-old person would actually be just entering the science. So we would say, you know, oh, he's only 30, he's a baby. So it's a lot about lifestyle, something about your genes, but it's something which you can measure, which can tell you how do you compare to other people. And the most importantly, this is something you can change. For example, my glycan age is lousy. I won't even say how high it is. Because I eat too much, I work too much, I'm too much stress, I'm horrible. But I was playing and testing, you know, I tried to sleep more, didn't really help. I tried to exercise more, didn't really help. So the only thing which works for me is losing weight. But I like to eat, so it doesn't work that well. So, but the thing is, you know, you have a tool how you can measure what actually works for you.
Spectacular.
I was talking too much. Maybe.
No, that was perfect. That's great. That's great. So it is affected by inflammation. So you mentioned that that's one of the markers which affects the age.
So it's not only affected by inflammation, it also participates in inflammation. So it's not that there is an inflammation which makes your glycans look old, but there's some kind of a signal, and this signal could be infection. You know, you can have I don't know, infection in your oral cavity. You have some kind of bad tooth or something like that, which is driving infection. You could have a chronic infection somewhere in your body, some kind of bacteria somewhere, or you could have inflammation which originates from a tissue damage. For example, if you exercise too much, actually you hurt your body and you initiate inflammation, which is not infection, there's no bacteria there, but there is a tissue damage, and then there is a repair process, and then the glycans play the role there. So glycans are part of the inflammatory process. So good thing about inflammation is there is a damage, you start inflammation, you destroy everything there, and then you rebuild it. This is the way inflammation works. So, you know, bacteria come in, then your blood cells come and kill all of them, and then you repair the damage. But if this happens too often, and now we are talking about this chronic inflammation, which can last for decades, then it starts to damage the tissue. Like in anything you're repairing, there is always a little bit of damage. So with too much inflammation, you kind of have a lot of scars everywhere. And then this is making you kind of biologically older. And we did another interesting study. We compared people in different countries. We compared 27 different countries all around the world. And in some of those countries, people would have something like, we even have a Papua New Guinea, where at that time, the life expectancy was something like over 40 years. And then we have some African countries and some Asian countries and most of Europe. And we could actually correlate the GlycanAge there with the expected lifespan. So if you are 40 and you live in a country where the expected lifespan would be 50, your GlycanAge would be very close to the maximum, which maybe in UK, people would reach in their 60s or something like that. So it really depends how you live and how your glycans will change through your lifetime.
That's really interesting. Ooh. So, well, I've hung around a lot of health conscious geeks, and many of them strongly believe that epigenetics tests are the gold standard of age testing because of its correlation to DNA methylation, possibly, or maybe other factors. But
so
how
DNA
would
methylation
compare? How would they both compare side by side?
So DNA methylation is something which is easy to measure. And this so-called epigenetic clock, which was developed by Steve Horvat, maybe even a decade ago, is really reliable.
2013, I think.
Sorry?
I think it was 2013.
Yeah, something like that. Yeah.
Hmm.
So it's something which actually very reliably can predict your chronological age. So the original epigenetic clock is so accurate that it is actually not really useful because, you know, everybody knows the chronological age. So if you guess it with a 1 year error, what does it help? And actually, people now use this Steve's original clock, which is measuring epigenetic methylation at something over 300 sites in the genome. Mm-hmm. They use it to estimate the age of the illegal immigrants. So, you know, somebody comes in as an illegal immigrant, says, oh, I'm 17, and they do the test, now you're 25, you're kicked out. and this is, and even I spoke with Steve about it actually it was even more than a year ago. And he's now developing some new tests because these are too accurate for the chronological age. So now he has something which he calls a grim methylation age, where he modeled how much you smoke and some protein biomarkers. And this is now kind of predicting the unhealthy life. So the key problem with the methylation is that you measure something like 600,000 sites, and then you can model more or less anything. And probably if you would take the same people, GlycanAge and methylation, you can make a methylation clock for GlycanAge. And this is when you measure everything. Now you have these commercial tests for methylation age, which don't use the original 300 plus sites because it is too expensive. So you cannot measure everything. So they measure 5, 10, 15 methylation sites, which correlate reasonably well with the original clock. But the key thing is what explains the difference. And so far, none of the commercial tests have actually shown what explains the difference between the real age and their epigenetic age. Because they claim, yes, with the Steve's clock, you can show, you know, you're a couple of years younger or older, or this associates with the risk. But for the commercial ones, still nobody knows. So it's any test you take is a kind of a gamble at the moment, because you will not get the same results from different companies because they measure different things and they're all hiding what are they actually measuring. So I'm a big believer in epigenetic information, because I think, you know, all our life events, experience is being actually recorded in epigenetics. And I believe big part of the glycan changes is recorded in epigenetics, but not in those couple of sites which are being sold as an epigenetic test. So I think the advantage of GlycanAge is that we are not measuring information, we are measuring effector, we are measuring the molecule which actually does something. And since we are measuring the effector, the change in an effector will have repercussions for health and disease. And this is what we are showing. So we already analyzed over 150,000 people in the different research projects we have. And in most of the diseases, we see that these glycans, which are basis of a GlycanAge test, change with disease, and often they actually change before people get ill. So
Right.
for example, we did recently a cohort of 27,000 people in Germany, which were collected nearly 30 years ago, and the blood samples were stored. We analyzed the blood from 30 years ago, and we knew who had a heart attack or stroke in the next 10 years after the collection.
Oh, wow.
And we did see that the glycans which we measure are predictive of who will have a heart attack or stroke in the future. So it is not that, it's not a diagnostic test. We are not claiming it to be a diagnostic test, but this is definitely something which is important for your health, which you can measure, and then you see what actually happens.
Wow. So back to the Steve Horvat test. You said the commercial ones only measure about 15 of the, is it genes?
So they're hiding the exact number.
Mm-hmm.
So they measure a couple. I know some of the companies which measure only 5. I don't know how many of them all the other companies measure.
Mm-hmm.
And they developed these few because they correlate reasonably well with the entire 300.
Right,
but,
okay.
As I said, the key thing is what explains the difference. Because the difference could be explained with something which is completely irrelevant medically. And so far, there has been very few tests which actually show that you can change your methylation age. There was one published last year with a very aggressive hormonal therapy where people actually got younger for a year or so. And there has to be a more research. Frankly, we don't know how things which we do change your lifestyle. And there are many people doing research, so there will be more and more data about epigenetic clocks. I recently spoke with David Sinclair. He has some interesting data about methylation also. We're also doing the glycans with David Sinclair, so we'll see how these two things compare.
Oh, amazing.
So, we are just collecting pieces of information, trying to see how they can actually help people.
So, yeah, I've been listening to an audio book by David Sinclair, and I'm not a scientific person, and I had to put it down because I felt like my brain was on fire. Seriously, obviously, this is his life. This is what he does. And the reason why I keep mentioning about epigenetics, because I know that there is definitely there's some substance there, and I just want to know if there is a particular way that glycans and epigenetics can kind of codependently work together, to get some results.
We are doing a couple of studies where we will measure both. And as I said, the key problem with epigenetics is that while we do this at the research level, meaning when I collaborate with Steve or David, then they would measure 600,000 sites. Wow. 600,000 is methylation points in a genome. And then we find which of them bring important information. And we did a couple of those studies, and there's always plenty of very important information in methylation. Problem is that if you just order a commercial test, they just give you a number which you don't know how exactly was it calculated. So this is why I'm not too enthusiastic about those commercial tests which are currently available. And actually, if you see, none of those companies really is very successful. While I am very optimistic about what we do with glycans, because we know exactly what we measure, we know what these molecules do, and we know that if, for example, we had a group of people undergoing bariatric surgery. So bariatric surgery is when they kind of reduce your stomach, so you eat less, and then people lose 20, 30 kilos. And we had a patient who was 30 years younger after 6 months.
Oh, wow.
It's a drastic therapy. You don't want to do bariatric surgery, but in 6 months, he got 30 years younger, one person. And we also had a large study on 2,000 twins, which were followed for 20 years. And we had samples in 3 time points across these 20 years. And twins which were actually losing weight in that period were aging much slower than the twins who were gaining weight. So, you know, if you're gaining weight, your kind of GlycanAge goes much faster than if you are losing weight. So it's fun doing research. So this is what I enjoy. You know, we are kind of learning what happens if you do something. We had an interesting cohort of ladies preparing for the bikini fitness competition in Finland. And actually, while they were doing this very intensive preparation, they got worse. But in the recovery phase, they got better again. And then we had another cohort of people doing high intensity interval training, and they got immediately better. So obviously, depending on what kind of exercise you do, your body responds in a different way. And I think the key question here will be is what kind of training and what kind of diet would be optimal for a given person, because we are all different.
Yeah.
But unfortunately, even if we sequence the entire genome of a person, we cannot say what that person should do. There is still not enough knowledge to interpret the genetic data. So the only thing we can do at the moment is try something and then measure how it affects your body. And if it works well, okay. If not, try something else. And this is what I was doing. So I was trying sleeping, didn't help. Exercising more didn't help. I have to eat less. Horrible. But at least I know what I can have to do if I want to have an effect. Some people could be lucky. You know, it could be only sleep a little bit more. I would immediately go for that option. You know, sleep 2 hours more and you'll be better. But I'm not the lucky guy.
Oh man. So we are all trying to live a healthier life.
But I think that's the wrong conclusion. Majority of people are not trying to live a healthy life.
That's a valid point, actually. Yeah.
The reason why they don't do it, because this is difficult and they don't see the effect. So living a healthy life will give you a benefit in 20 years. You know, you can always wait for another week or a month or a year before you start investing for something in 20 years. And there has been a research showing that if we just get rid of the most important unhealthy habits, we can add 10 years of healthy life to our lifespan. Amazing. So we can really kind of save 10 years of healthy life, not any life, healthy life. And people don't do it because the reward comes too late. You know, I'm not going to sacrifice today for something what will happen in 20 years. And the idea of the biomarker would be that you can see effect immediately, not immediately, but in a couple of months. And we have a number of people who, for example, they would do the test and they don't look that good. And then they start thinking and they change something. And if they see an effect in 3, 4, 5 months, then there is how, wow, there is an effect. I'm going to try harder. I will keep doing it. While if you don't have a feedback, you know, then you don't do anything. So I think we cannot extrapolate ourselves. So there are many people who really think a lot about their health, but the majority of population actually don't.
That's interesting, isn't it? Like you'd think that everybody has a mutual interest. I think people don't necessarily do their best to be healthier, but they just kind of expect it to happen. They're like, I want to be healthy, but it doesn't mean that I'm going to do what it takes to be healthier. I think people just want to live long and they kind of want to thrive without doing, you know, the effort that's required. As glycans is, I think it's one of the 4 building blocks, isn't it, of life, alongside protein and nucleic
acids and lipids.
Yeah, and lipids. How would you specifically make your glycans really thrive? You know, what is the ultimate measures, you'd say?
We don't know. So
Yeah.
what we know, so first, the glycans is a very broad word. Like, proteins, nucleic acids, lipids is a very broad terms. GlycanAge is more narrow. So GlycanAge is measuring glycans on immunoglobulins, and this is related to inflammation. We also have some other glycans which we measure, for example, related to more metabolic health and so on. So GlycanAge is mostly related to inflammation. And what we know that what we can do for this GlycanAge to be better is, one thing is definitely get rid of excess of food what we are eating. Eating too much food is definitely harming. And also other things which are considered to be unhealthy, like smoking is an important factor. And stress, psychological stress is an important factor. Physical harm to the body is an important factor, and you can actually even harm yourself by exercising. So kind of if you are wearing out your body too much, you can see that. And this is something we see with the professional athletes, that it does move faster. But the good thing is that damage can be repaired. Mm-hmm. We had, actually, this was even published by Daily Mail, I think a couple of years ago. We had a person who was living a very unhealthy life, but took an early retirement and started living much healthier. And actually, he went all the way below his chronological age. So no, these things, this is not that you are damaged forever. These molecules can repair themselves, and doing the test can be kind of an early warning. There is something wrong, and I can actually repair the damage before the damage starts doing the real harm. Because one thing I did not explain. So people usually think either health or disease. It's kind of a categorical term. I'm healthy or I'm ill. And this comes from a modern medicine, because how do we define ill? You go to hospital and then you get a diagnosis. Somebody tells you, you have a diabetes, you have a hypertension, you have a cancer, whatever. And this is the moment when something is so wrong in your body that it doesn't work anymore. So you feel pain in one of your organs, or glucose is no longer regulated. So it's a kind of an endpoint. And between this endpoint, which we call disease, and the healthy state, there's a kind of continuum of damage. So our body can compensate most of the damage. So something happens, you compensate. Something happens, you compensate. Only after a long time, you decompensate, and then you're ill. Mm-hmm. The idea of a biomarkers, like GlycanAge, is that you identify this disease developing process before the endpoint. So you don't wait to become ill to go to hospital, because once you are really ill, modern medicine rarely cures the problem. Modern medicine just reduces the level of symptoms so you can function. But there are very few examples where you can actually go with the disease, maybe, I don't know, some hematological cancers. You have it, and then you cure it. Most diseases which are burdening the modern society are chronic diseases. So once you are ill until you die. It just gets worse a little bit. So with biomarkers, you can see this process developing. Try changing something which might be really easy. For example, if you develop diabetes, you really have to change your lifestyle. But maybe if you identify the risk 5 years earlier, maybe you have to change your lifestyle only a bit. For example, something which really seems to be working is this intermittent fasting. So just, you eat the same amount of food, but you limit it in a number of hours per day when you eat it. This seems to be working. Or you think more about your microbiome. You eat a food which your microbiome likes, and you can prevent the disease.
I think a few questions was coming in my head, and as you started talking about microbiome, I was going to ask you what's your thoughts on Like you mentioned about eating food which is good for your microbiome, but we all have different microbiomes. It doesn't respond the same way to every type of food. And what's your thoughts on having a test for that? Do you think that they are accurate enough to tell you what is the right food for yourself?
So we still know very little about our microbiome and how it affects health. And I think the only thing where we have kind of a consensus is that a good microbiome is a diverse microbiome.
Right.
So the measure of the microbial health is how diverse it is. So when I said a good food which is good for a microbiome would mean you have to eat diverse food, especially many different plants. For example, Tim Spector, who is one of the leading researchers in the field from the King College London, says you have to eat something like 30 different plants a week.
Right.
And then if you eat sufficient diversity of plants, then your different microbes can thrive. And then by having a diversity, they can protect you from all potential invaders. Because the problem with the microbiome is that we have a kind of friendly microbiome, we live with it. And then if a pathogen comes in, I don't know, you eat salmonella or something like that, then our friendly microbiome will fight them and will not let them make us ill. But if we have a very unhealthy microbiome, for example, again, with the Tim Spector, I think his son Tom was eating McDonald's for a week or 2 weeks, something horrible.
Wow.
And this reduced his microbiome drastically. And then when you only have few bacteria growing yourself, then maybe one which comes in can make a chaos and make you ill. So the idea about the microbiome, and we know that microbiome affects also the glycome, is it should be diverse. We also did an interesting experiment. We did, it was done, I think, in Canada, that people were getting so-called fecal microbiome transplant. So you take a microbe from guts of one person and then transfer it to another person. There are several ways. You can even just have a pills and then drink these pills. Replacing the microbiome, the unhealthy one with the healthy one, then also changes your glycans. So by doing the microbiome transplantation, you can improve your glycome.
That's really interesting. Really
That's
interesting.
a classic approach. Maybe just a healthy food would be.
Yeah, yeah. But the diverse food is really interesting. What I find is I try to keep it as diverse myself.
One more thing which I didn't mention. In addition to being the diverse, one of the key problems is so-called ultra-processed food. Because now we try to invest as little as possible in preparing food. So we are buying food which was prepared somewhere else. And the problem with that is that these companies which are making food ready for us, they have to keep this food, it has to stay functional for weeks practically, even more. Because logistically you cannot make it every day. So they have to make something and then ship it to the shops, and then people buy it, store it in a fridge. And what they do, they put the different conservants, different chemicals, which prevent bacteria from growing. And all the semi-prepared or fully prepared food that you're buying will have different chemicals which kill bacteria.
That I understand.
Chemicals, you kill your own bacteria. So you're killing your microbiome with the food you are eating.
Yeah. What's your thoughts on seasonal eating? Like some people believe, like, eat certain foods which are in season, and there's lots of fruits, for instance, which are shipped into other countries, even if they're organic. What's your thoughts on eating stuff which is not in season?
This is not really my field, so I'm not an expert in this, but
right.
I just say it logically, is that the food, the fruits, the vegetables which have been shipped haven't been really damaged. You haven't done anything bad to them. But I think the problem there is more that the quality, in a way of first the taste and then maybe different nutrients which could be there, will be very different, because all those fruits which are kind of selected for keeping them for a long time have been selected for this. And if you compare any fruit which you actually climb a table, climb a tree and pick it and eat it, it's uncomparable with what you buy in shops. So
Right.
I don't think there is a huge risk in doing that, but there is a huge loss in quality because, you know, I do have some fruits which grow in my garden and it's uncomparable to what you buy in the supermarkets.
Yeah, wrapped in plastic as well, isn't it? So you briefly touched on the exercise part, and you'd mentioned that some training can cause inflammation in the body, and then our body can recover from it. And I think you mentioned high intensity training showed a good response to glycans, or glycans responded well to the training. Would you say that high intensity training might be best overall, or it just varies per person and you got to check it out yourself? See if
We don't have sufficient research yet to say
right.
that. So we did 2 small studies. We have a couple of hundred people on the different exercise regimes in the moment. We are doing another study. And I think there is definitely an advantage of high intensity training because there is a recovery period. So I think recovery is something what is very important because if you have a continuous work being done by muscle, there is no way to get rid of all those molecules which are kind of byproducts of metabolism. And then we all know that if we don't exercise and then we start exercising any kind of specific muscle, then we feel pain in that muscle after a few days. This is because there is inflammation going on there. And this inflammation is something which is definitely not good. So this burst of intensive activity with recovery period seems to be beneficial. But then again, I'm a biochemist. I'm not a specialist in different forms of physical activity. So, and this is based on the limited research we did. Something which is definitely needed is to see how different people respond to different regimes, because we know there's some kind of genetic predispositions for a different type of physical activity, and then combining some kind of genetic data with different type of exercise could be beneficial. But for the moment, on a planetary level, the level of knowledge is too low. We have to do much more research to understand. And every model which claims that, you know, something is good for everybody, this is wrong. You know, people have to see what actually works for them. And, you know, the best way is, you try it, you measure it, and then you see what works.
How do people measure, you know, their glycans on a regular? How often would you suggest the person measures it? Like, you know, they try a different training program or diet, and yeah, how often should they measure?
So glycans change relatively slowly. So unless there is some kind of a really drastic thing, like kind of acute infection or something like that, it takes at least a couple of months for them to change significantly. Personally, when I track myself, I do it monthly, but, you know, I have a lab who's doing that. I don't have to pay for that. So too much, but something like every 3 months or every 6 months would be something which would be informative, because in 3 months we would see effects of kind of intervention we are doing.
Cool. And where do you see GlycanAge moving forward? Are there going to be any other things added to it to help to improve the science?
So we are doing the research all the time. So personally, I'm running a research lab. We do 30,000 people a year, different glycan analysis, and we are learning more and more. So I see GlycanAge eventually moving to kind of routine diagnostics, but this will take probably a decade or so. For now, what we are doing, we are offering this test to people. They can try it. We are also offering it to different clinical partners or different lifestyle coaches, where people use they use our test to evaluate interventions they do, like different longevity centers or something like that. And the way I see it, people will start using it more and more. We will learn more. We'll be able to give more personal guidance, which we cannot at the moment, because it's not based on glycans. We cannot say what will work for you. You know, we can only say in different people, we know that these things might work. For example, if I look at you, you're probably not overweight. So I would not tell you, you know, lose some weight. It will have to be something else. So yeah, we'll just learn more. And as we learn more, we'll be able to have more fun.
Fantastic. Fantastic. Sounds good. Sounds good. Where can we find more information about yourself? Like, have you got any blogs or just more like research stuff that you're working on?
I regularly post what we do on Twitter and LinkedIn.
Mm-hmm.
So there are many posts by me. I nearly post every few days. And this is where I usually try to translate what we do into the lay language. There is a knowledge hub at the GlycanAge.com site. And this knowledge hub, we try to explain not only the GlycanAge, but other aspects of glycosylation. For example, now we have a lot of data about the COVID, because glycans are also important in the COVID, and everybody is interested in COVID. So the first thing would be look at the GlycanAge.com, check our knowledge hub, and follow me on Twitter or LinkedIn.
And what is do you have a personal Twitter and LinkedIn?
Twitter and LinkedIn accounts.
And what's the handle? What's the name?
It's my name, Gordan.Lauc, something like that. Sure.
Okay. Well, if you could just send it to me, I'll put it in the show notes on the podcast.
Yeah, it's just my name, so it's I have a unique name on a planet, fortunately.
That's cool.
Yeah.
Awesome. All good. Thank you very much for your time today. That was some golden information. Yeah, it's been an absolute pleasure. You take care, sir.
Good.
Okay.
Bye.
Goodbye.
So I'll send you the link the moment I get it.
Yes, superb. Thanks a lot, man. Thanks a lot.
Let me know once it is up, so I'll also share it on my social media. I don't have so many followers, but I have combined Facebook, Twitter, LinkedIn, something like 30,000, which is not that bad for a scientist.
Oh, that's pretty cool. That's pretty good. Pretty good. And they're all dedicated people, so that's going to be great. Now, I look forward to sharing this. There's a lot of people that, you know, as I mentioned already, a lot of people know about the whole epigenetics. It's almost like that's been given a lot of, I don't know, a lot of presence online.
I didn't want to say it in something what you would publish, but the real problem is all those commercial tests are actually cheating, because they made a cheap substitute for the original test. They say it's based on the Steve Horvat's epigenetic test, and they deviate from the test between 5 and 10%. And this difference is much larger than what is the link between the real test and any kind of health effects. So I'm afraid that most of those commercial tests, when compared to the real outcome, it will just be random. Yeah. So I'm afraid there will be a big bad publicity once this come out. But
no, people need to know, because yeah. That's cool. That's it's good to know. Because people are spending a lot of money on these things. You know, I've been lucky in a sense where companies have come to me saying, like, you know, would you be interested in, you know, taking a test? I'm like, yeah, of course, I'll take lots of tests. Why not? Learn a bit more about myself.
Didn't we? Nicolina, I think she made it for you, right?
Was she done a test for me?
GlycanAge. Did we do?
Yes. Yep. So I'm currently 58 years old. I'm not too bad for 58, am I?
I'm 75. I'm not 75.
You're 35.
I'm 75 in GlycanAge.
You're okay.
Yeah. So it's well, it's a lifestyle. My lifestyle is horrible.
Right.
So what's wrong with you? Were you ever for how long you really seem to be kind of fit?
Well, You know, that's there's another guy who I'm going to be speaking to, some professor, where I'd be able to hopefully get some answers, find out where I've gone wrong. You
How
know,
often do you exercise?
well, to be honest with you, not as frequent as a lot of people might think. I used to train 5, 6 times a week, but that was a very long time ago. I think at that time, that was probably about 5 years ago when I was competing. But like, you know, I'm doing a lot more things to improve my health on a cellular age now. So I'll do things like, you know, I'll have like molecular hydrogen water. I'll have, I'll do like photobiomodulation, also a bit of light therapy. I'll try and stimulate the vagus nerve at nighttime, breathing exercises, and I'll train probably twice, maybe 3 times a week, not even as aggressively as I used to. So
How old are you?
there's other stuff. You know, I'm doing intermittent fasting as well.
How old are you chronologically?
I'm 41.
So this between 40 and 50, men don't change that much. So the fact that you are, you know, maybe the next test you could be 5 years younger or something like that. And the other thing is that you have been, we have only white British baseline. And you obviously have some other genes than white. And this genetic background does usually associate with a slightly higher GlycanAge. So
Right.
maybe, I don't know, I guess your family lives in the UK for a long time, right?
Some of them, yeah. Mm-hmm.
No, so your genetic origin would be from where?
From Caribbean. Well, genetically, it says Africa because I had one of those, but like my parents are from the Caribbean.
So your parents already were living at Caribbean. They moved relatively recently to UK, right?
And they moved to, it's been, well, as old as I am is when they moved to UK.
I'm talking now evolutionary, so this is recently, you know, 80 years. Yeah, so I guess if we would plot you, we have, I think the only country we have is Trinidad. Let me check Trinidad.
Right. My father is from Trinidad.
Yeah.
But you know what?
Stay with
I'm
me
gonna
for a
have
second.
a
Let me show you how Trinidad looks like. Just a second.
All right. I need to message someone because I have a call at 1 o'clock.
Okay, so so I think Trinidad baseline would make you at least 5 to 10 years younger. So based on Trinidad baseline, you would be approximately your chronological age.
Right.
Genetically. So I don't think you're doing something really wrong with your life. It's just combination of genetics and maybe too much sport, maybe when you were younger. Yeah. So
Right.
it's not that bad. So I wouldn't be concerned.
Mm.
Okay.
Thank you very much. We'll talk soon. Thank you. I'll let you know when everything is all up and running. And yeah.
Thank you.
Thanks
Bye.
again. Cheers. Goodbye.